So how's it going, everyone? So we're going to be talking to you today about the rescue transformation of yeast uh, with this plasmid CM184 and BCL2. I'm Justin Houts. I'm here with Christian and Sheena. We're part of the Fresno State Undergraduate Studies Department of Chemistry through Chem 156. So a little background of what we're doing here. One proposed treatment of cancer and leukemia is biochemical inhibitors. One issue in cancer cells is their fast growth and reproduction, so we can inhibit this process. We could possibly help them. Our goal is to use the yeast as this model organism to validate factors that are involved in the transformation process between yeast and the BCL2 family proteins. BCL2 is known to be associated with cancer, and so we've introduced BCL2 into a model yeast that is completely devoid of it. So this way there's no background responses or anything like that. Moving on, some of the other things we're looking at. So the BCL2 family proteins can control cell death or apoptosis by direct binding interactions. The BCL2 family proteins either promote or inhibit apoptosis, and this is done through mitochondrial outer membrane permeabilization, so they can make the membrane more or less permeable depending on what they're trying to do. However, the mechanism of transformation of BCL2 in cancer is still up for debate. So we're looking at the potential role of BCL2 at the metabolic level of yeast and to testing the impact of BCL2 expression on yeast metabolism.
So for our methods, the same shuttle plasmid PCM184 BCL2 was used for both bacterial and yeast cells. The plasmid contains genes for both tryptophan 1 and ampicillin resistance. Our bacterial cells, HB101, were transformed and grown on selective media containing ampicillin. This allows us to only grow cells that we want. It allows us to amplify the plasmid for later use in yeast cells. Our yeast cells, strain W303-2N, were transformed and grown in media void of tryptophan. By doing so, only transformed cells are able to grow. Yeast does not contain any BCL2 family proteins, making this a good control. Next.
For the results section, we have our transformation slide. On the left is our transform E. coli colonies. We see target colonies growing on the plate containing ampicillin and LB. There are also various smaller opportunistic, which are non-target bacterial colonies for our experiment. On the right are the transformed yeast cells. The yeast transformation produced very few colonies, but it does indicate that the transformation was successful since yeast is growing even though tryptophan is not present in the medium. Continuing with our results, we have our fingerprint data. The plasmid restriction map is on the right and the DNA fingerprinting is on the left. The restriction enzyme used was BAMH1, which performed 2 cuts on the plasmid. They can be seen on the image provided. The cuts were done roughly around 2500 nucleotide and the 3100th nucleotide, giving us roughly a 600 base pair chain, which can be seen as indicated by the red arrow on the fingerprinting image provided. All 3 of the samples have bands between the 500 and 700 nucleotide range, which is exactly what we wanted to see.
Yes. So for our yeast transformation, we were not able to produce as much colonies, but for the ones that were shown on the agar plates, it's possible that we could use it for other proteins. So for the future work, we decided that we want to do it on spotting assays. The inducing media could be used to change the cellular functions to enable the effectors to be more susceptible. So in other words, like we want to promote our growth for our protein. Next. So we would like to say thanks to Dr. DeJohn for guiding us through the experiment, our fellow classmates from Chem 156, along with the Fresno State Chemistry Department for lending us tools and allowing us to construct our experiments, and along with Dr. Manum. Thank you.